Welcome to Cancer Research Demystified. It's 2020, so Susan's not with me because at the moment we're locked down in our houses because of the coronavirus pandemic, which means this episode's going to have a bit of a twist. It's going to be about the coronavirus tests. So although we both do cancer research, there are some things we could possibly do videos on that will be of interest to people who want to know more about the coronavirus pandemic. In particular, one of the things is the tests. Now, the technology behind the test is called the PCR reaction, or the PCR test. It's short for polymerase chain reaction, and we use it all the time in cancer research, like constantly, and so do loads of other disciplines. It's a really fantastic way of testing things, and I'm going to go into a bit more detail. So the PCR test is designed to pick up bits of what we call nucleic acids, and that covers DNA and RNA and some other things that you have in every single cell in your body. But it's not just in your body, it's in the cells of plants, and it's also in viruses. So there's a lot of differences in DNA between all the different types of living cells in the world, and that's the same with viruses. There are some bits of that DNA that are very specific to the virus. So if we can use the PCR test to see those specific bits of DNA from the virus, we can see the virus, and that's how the test works. So the viruses are in you, that in your body you have a current infection. But you've also got a lot of other DNA in there, and you've got a lot of other cells in there. So what we do is they'll take a swab, and I think the classic one, especially in this country, is up the nose or down the back of the throat, and then they'll look in there to see if they can see bits of this DNA from the virus. So the technology this test is based on is absolutely incredible. So every single cell does this naturally all the time, and there's a little protein in there that's able to make copies of, say, a specific bit of the DNA, and it will just make some copies when it needs to, for if your cell needs to react a certain way, or if your cell wants to make multiple copies of itself. And there are slightly different versions of these, like enzymes in your own cells, and what people with the PCR reaction did was re-engineer one of them to be particularly useful for us. And what we can do is we can design little, what we call probes, against the bits that we're interested in. So in the virus's case, it will be the bit of DNA that's only specific to that virus, and tell this protein to make copies of it, loads and loads and loads and loads. So instead of that tiny little bit of DNA being mixed in with all sorts of other things, it makes so much of it, there's loads of it, and we can finally see it above the background noise of all the other DNA from all the cells in your mouth, from your nose, from wherever they've taken that swab, and we can finally see if the virus is there or not. So let's say this is the virus, this nice yellow star. Inside that is the bits of DNA, and there's that yellow piece of DNA, which is the bit we specifically know is only in this virus. Problem is, it's surrounded by cells that are full of all sorts of other bits of DNA, and when this all breaks up and gets mixed together, how are we meant to see our little yellow bit? What we do is we design a probe that matches the bit of the DNA we're interested in this case, the viral DNA, and then we add our clever protein. These proteins are called polymerases. That only sees this double bit of DNA where we've mixed our probe in to match it, and it can recognise this. What it does is it replicates it, so it makes multiple copies. So there is a bit more to the reaction than this. There's a lot of conditions we need to change, temperatures and different things we need to add in order to make the reaction work and actually this copying to happen. But this is the basics of it. So it does this again and again and again, and it'll keep going. What we finally get is loads and loads and loads, and it's hundreds of thousands times more copies of this particular bit we want. So much so that there's so much more of it than any of the other DNA that was in there. The extra clever thing they do is they add in a fluorescent dye. So this dye binds to the DNA, and once we get to a certain amount, we can see that dye and we can see it glowing. And this is where we get the positive test. In comparison, there'd be a negative test where that probe doesn't bind to anything, that matching bit of DNA isn't there, the polymerase doesn't copy it, and we end up with nothing and therefore no glow. So in cancer research we use a similar thing. If we're looking for some DNA or RNA that's specifically associated with cancer, we can look specifically for that in various samples. So we might take them from blood or urine, or even from the tissue themselves. So when you get an infection, you'll start with a little bit of virus. That might then build up in your body. It'll start to grow, and we'll then start to be able to detect it. Your immune system will then it'll start biting the virus. The amount of virus will then drop, and we might stop being able to detect that virus. So a long time after your infection, these tests won't work, which is why I want to bring up the other tests that you might hear the government or various things on the internet start talking about, and that's an antibody test. So the antibody test is different, and it's based on a technology we also use in cancer research. A lot of these big scientific discoveries and technologies are used all across different disciplines of science. The antibody test is where they're detecting the antibodies that your immune system produces when it responds to the virus infection. All these antibodies are different, and they respond specifically to the virus, so they might look slightly different from antibodies that respond to, say, a bacterial infection or something else in your body. It is a bit complicated with antibodies. There's different types that are around for different amounts of time and at different levels, and you can detect them different times after the infection. Here's a picture I stole from someone on Twitter, and I'll put who posted it. On the bottom here is time. The first peak is the amount of virus you have. So it's where this PCR test that I talked about earlier will come in useful. So you don't have any detectable virus, you'll have a lot more virus, your immune system kicks in to kill it, and then the virus drops. This second curve is the first part of your immune system, that really big boost that tries to get rid of the virus. And as you see, this goes up, and it comes down again and tails off. But you get a prolonged immune response where your body goes, I might need to remember how I killed this virus, and it produces the second type of antibody that helps you remember you've had that infection, and if you have it again, respond quicker. So you can see these different types of antibody that your body produces to fight the virus come up and go down. So you can develop tests against these different types of antibodies, and these tests are being tried to be developed at the moment. But it does mean this antibody test means we can pick up if you had an infection in the past, which will be very useful in the months to come. Antibodies are used all the time in cancer research as well, in a number of different ways, and we can do more videos about them. They're again an example of something that the body does automatically, and it's incredibly beautifully designed. And we just abuse that a little bit and use it in science to find out more about what's going on. So, a bit of a spin-off, a bit of a coronavirus-style cancer research demystified. Thought I'd do something a bit more coronavirus-y, since that's what everybody's talking about at the moment. If you find this in 2021, 2022 and everybody's bored of coronavirus, just skip. We've got more amazing videos about cancer research. You'll find them in the list or in our stories or all sorts of things. So thank you for listening, and next time hopefully I'll be back with Susan.